All right, we're ready to get started. Thank you. Great. Stand on my tiptoes. Good morning, everyone. It's great to see everyone here bright and early for this first session of the plant section of the conference. Thank you for being here. My name is Joyce Van Eck. I'm a professor at the Boyce Thompson Institute, and I'm also the chair of the steering committee for Plant Gene. So I have just a few slides to give you some background on this session and some logistics about how we're going to work it. It's a combination presentations and panel discussion, and we're going to ask you for your participation as well. So the Mastering Plant Transformation Vector Design, the way this came about was feedback from the community. We asked, at our last workshop, we had a survey last year and asked what people were interested in, and people wanted to know the basics of vector or construct design, and that's what we're going to do for the next 3 hours. So I've listed the presenters here. The first presenter will be Dr. Vina. Oh, I have to click that, sorry. I'm so used to using my computer. Thank you, Vina. The first speaker is Dr. Vina, and then Dr. Kun-Sub Lee. And you might be wondering, this is a vector design. Why are we talking about agrobacterium and the basics of tDNA vectors? Because that's really important, and you'll learn why that's important for when you're choosing a vector and designing your constructs. And then Katie Toomey will talk about cloning and vector construction basics. And then we'll end by the Plant Gene Steering Committee talking about tDNA design and cloning tips. Oops. Okay. Hit it one too many times. All right. So for the logistics, we are going to have 5 minutes for questions after each presentation, but there's definitely going to be additional time after all the presentations are complete. The last presentation will be on tDNA cloning and tips, and this is the interactive part. We're seeking input from all of you on the different tips that we're going to share. And then at the end, stop by after the workshop and for a pin if you're a Plant Gene member, and Amy has all the pins, and you can get one. And if you're not a Plant Gene member, you can become one. You can scan this QR code. It'll take you to our website, and there's a form to fill out to become a member. And we have some of the resources that are listed here on the website. And I'd like to thank the National Science Foundation, specifically Diane Okamura, for helping with SIVB and getting a grant to support the workshop so that we could have this recorded, and it'll be available on the SIVB site and then also on the Plant Gene site, and you'll get emails alerting you when these recordings are available. And also to SIVB for allowing us to have this platform to host the workshop, and to all the Plant Gene members for your engagement. All right, so thank you. And we're going to start with our first speaker. Bill Gordon-Cam, who's a member of the steering committee, is going to introduce Vina.
So it's a great pleasure for me to introduce the first speaker this morning, Dr. Vina from the Danforth Center. Vina is a real expert in agrobacterium. She did a postdoc under Stan Gelvin for around 4 years, roughly 20 years ago or so, and that gave her a lot of really great basic background to the topic. And then since then, she's been learning more every year about the practical aspects. You never stop learning about agrobacterium. So all of the people in the audience that have been in the field for 30 years or more, I'm sure are still learning about agro just as I am. So Vina is currently director of the Plant Transformation Core Facility at Donald Danforth Plant Science Center. She does both research and service transformation. Her research centers around novel methods of transformation and genome editing. And as the large part of the core facility is focused on client satisfaction. She works with both academic and commercial labs around the world to satisfy their needs and improve crop performance. So I will let her take it away.
Thank you, Bill, for introduction. He was trying to imply that I'm very old, and so I want to say that. And then agrobacterium is really, really old. And then while preparing this presentation, I was looking for historical information. And as Bill mentioned, I did my work with Stan Galvin as a postdoc. And I learned about agrobacterium and why it matters and what you could do with that to make the transgenic plant and why it is important to understand molecular basis of it. So my knowledge about that came from Stan. Tan Wang is in audience and many others, including Chuck. And then Stan Galvin is going to be here as well for 2 or 3 days. And so feel free to talk to people who have done a lot of pioneering work in this field. And just like you, many of you, I was the user of this technology. And then I got frustrated that why it takes so long and all those things. That's how I chose to understand more and work on that. So that's how I can leverage it more effectively. And so with that, I'm going to start my presentation. And I would like to thank NSF for the funding and then PlantGene platform. And it has been phenomenal experience working with people and all of you who have expressed so much interest attending these workshops. And I can't see my slide very well from here, so if something doesn't advance, please say that. And so as we all know, it is a gram-negative soil bacterium. It is a phytopathogen, infects plants, and causes disease. It causes gall disease, and then depending upon the way galls look, it can be called ground gall, hairy roots, or cane gall, and so forth. So this is the picture I took at Purdue University. So if somebody goes to Purdue University, they can look for a plant tree, look like this. And so it's a crown gall on a tree, which is caused by Agrobacterium tumefaciens. And this is the next one image is the hairy root on the tomato in a hydroponic setting. So it's a real problem. And so why people started studying? Because it caused disease. And while studying that, people realized its potential to transfer the DNA into the other organism. That's what we are utilizing for the genetic engineering of plants. This is what is very fascinating to me. When I started working with Stan, I thought it is like 1900 or something. People started learning about crown gall and the disease and the agrobacterium, but it goes back to 1650 when somebody really reported in a literature the observation of disease symptoms in a plant. And since then, how far we have come. And as you can see, the major breakthrough in this technology and potential of agrobacterium, it happened. That is called the golden era. Like if I have to say, it's not major breakthrough, it's a golden era where a lot of things happened, where we understood that how tDNA is transferred, and then utilization for the genetic engineering of plants. And why I think this publication is very important, it is like not very long publication, so you will not get bored, but it will give you the references. So I would encourage everybody, like whosoever is interested in learning more about the history, please go and read this publication. And then the way it is laid out, there is a name like year when it was reported in the literature and the lead scientist. And this is what, breakthroughs happening in this period of 1970 to 1993. And so I really like this publication. Oh, sorry, I am seeing that, but sorry. So I just pointed this out just for the sake of knowledge and where to find this information. And then what I also want to mention that when these things, research was happening, that time a lot of molecular tools were not available. Sequencing has become very accessible now. So what has people were able to do without those capabilities? And so now if we apply those, we can do much more. And so many times I hear people talking about, oh, there is no more to be done in the field of agrobacterium. But more I go through my slide, you will realize that how little we know and how narrow focus we have. And so hopefully some of you, it triggers some excitement and work more on this field. So this slide talks about the phylogeny of the agrobacterium. It belongs to family Rhizobiaceae. And then many times you will hear the word agrobacterium or rhizobium. And then people who know about how the classification was made, it was debatable and keep on evolving, keep on changing because the uniqueness of the agrobacterium and how people see it causes the disease, what are the factors or the DNA regions of the agrobacterium cause the disease. So it keeps on evolving, and this is the latest publication I could find, and so that's why I'm basing this information here. And so there are different types of agrobacterium which were found in nature, and then these agrobacteriums were isolated from the infected tissue. The third column in there shows whether they are forming or known to form the tumors or not. And the red arrow points towards the commonly known agrobacterium we work. But then, I do want to say for the beginners, you can think about, there are other forms of agrobacterium you could use and explore. And on the right side is the phylogenetic tree, and then again the red arrow presents where the agrobacterium sits on that tree. So again, how different they are, and so there is a potential, again, different agrobacterium strains can expand the host range. And then agrobacterium in nature is capable or known to infect dicot plants. But under laboratory setting, it can infect many other organisms and plant species. With that, I'm going to switch gears and talk about the agrobacterium tumefaciens genomes. I chose C58 because it's the commonly used strain and then one of the most studied agrobacterium strain. And this slide depicts the wild-type agrobacterium strain, and we are going to walk through this. And the reason I'm explaining this is many times when we think about the agrobacterium, we just think about the TI plasmid, nothing more, as if nothing else exists there. So there is a TI plasmid. This is what we are used to know and work with. And in these agrobacterium, if it is a disarm, we would put the other plasmid. But right now, we are just thinking about this as a wild-type strain, which is capable of infecting or causing the disease in plant. So there is a TI plasmid or tumor-inducing plasmid. There is a circular plasmid there, which is P80, Agrobacterium tumefaciens C58 plasmid. There is one linear chromosome and then one circular chromosome. So this is all inside the agrobacterium. Oops, sorry. Okay, this is what. And so, so people used this strain and modified it. And then this circular plasmid, the TI plasmid, is capable of transferring to other agrobacterium. That is what, changes what the agrobacterium can do. And during the, how people started modifying these agrobacterium strains, there were different technologies were used, different resistance marker were placed in different regions of the chromosome or the TI plasmid. So it's very important for you to know what you are working with if you are troubleshooting. And then especially when we have so much knowledge about the sequence of this strain, then it will confuse why it is there if you don't understand what is there to begin with. So we know that agrobacterium infects plants, and when agrobacterium interacts or come closer to the plant cell, then there is a signal happens, plant-derived signal or environmental signal that would induce the agrobacterium proteins, and that lead to the transfer of the tDNA into the plant cell. It's very important to understand and think about these principles because that's how we can troubleshoot the efficiency or efficacy of the transformation process. This slide focuses on the proteins or the genes which are from the agrobacterium itself, and then how it plays a role in the transfer of the tDNA into the plant cell. So as the first line, like here we say, like wounding or environmental signal activates bacterial virulence machinery. Then there is an induction of virulence gene. It processes the tDNA, which is a single-strand DNA fragment, which is very sensitive to, breakdown. So there is a lot of proteins come into play, including BD2, which is a pilot protein, which guides the tDNA into the plant cell. And there are many other proteins in there which has different roles to protect the tDNA, help its movement from agrobacterium to the plant cell, and so on. And then once this all protein and tDNA moves into the plant cell, it eventually integrates into the plant genome. And so what we are again seeing here is the proteins and the genes involved from the agrobacterium playing a role in the integration or transfer and integration of tDNA into the plant cell. There is a whole lot other world, which is the host site. And then this world of host site changes depending upon the explant type, the plant type, and so many other things. And so there are thousands of genes induced early on or later during the integration process that determines the successful outcome. And to name the few, there is a plant defense response, and this is the publication you can look at, and then references therein to find more information about those. And then it is very obvious, plant pathogen interaction happens, there will be defense response. There is a tumor forming, capacity of the agrobacterium. So there is, transporters, development genes, and DNA repair machinery because there is a integration of the foreign DNA into the plant genome. So it's kind of very obvious, but there is a fascinating literature about that people can look into, read into, and then if you understand this more, you can again set up your optimization studies according to those understandings. So today's session is focusing on the vector design. And so these vector designs were founded on the principle of what is the tDNA, what it is TI plasmid, and what it is doing. So we thought it is very important to give the basics, especially for the beginners. If you are expert, please pardon me, and so ignore this. And so the TI plasmid have various components. So these are, it is a 200 megabase pair, 200 kb size plasmid. So there are a lot of genes, but we are just highlighting few regions here. There is a tDNA region. In wild type situation, it is consisting of, tumor inducing principle, which will be hormone regulation gene. Depending on if it is a RI plasmid, there will be different sets of genes which would cause the hairy root formation. And then this one is flanked by the left border and right border sequence. There is a virulence machinery for the gene for that, the replication for this plasmid. Opane synthesis, so many of you may not know because most of us do work with disarmed plasmid, and so you don't care about the opane. But agrobacterium used this, had this thing so that it can make plant to produce its own food. And that's what so there is an open catabolism genes that's here, and then there is a conjugal transfer gene there. And so one of the thing, again, these genes, the conjugal transfer genes helps in mobilizing the plasmid to other agrobacterium strains. So that's how people were able to make modification to the agrobacterium, disarmed agrobacterium strain. So what is the disarmed agrobacterium strain? It is the TI plasmid, agrobacterium strain containing a TI plasmid which is without oncogenes, and or the TI plasmid without any tDNA or the border repeats to it. So if this is what the wild type TI plasmid looks like, so disarmed would look like this, just to simplify the this slide shows various types of agrobacterium strains, which has been known in literature. Again, this is really good publication people can look at. And when we see this in the first table I showed, where I showed different types of agrobacterium strains, and this is the commonly used for the genetic engineering of plant or the basic research in agrobacterium. So it looks like quite a whole lot, but many of those are derived from C58 strain of agrobacterium. And this particular publication shows real good lineage of how things were evolved. And so they did pretty good job, and then color coding, and then resistance marker, and where the resistance is sitting, and all these things is really highlighted well in this publication. So I'm not going to go into detail, but you can see there are, how for example, we'll use BB3101, so it's derived like that. And how exactly, everything is being evolved. So one strain, if it is not working for you, try other. And then, so this is what, like, the key takeaway message I take from this, that there are different strains, and then if you are struggling, know what strain you are working on, and then explore from there. Next presenter will be talking more about the tDNA vector system, so I'm just touching the higher level view of various types of vector system. There is a co-integrate system, there is unitary or binary vector, ternary vector system, and then multiple tDNA. So I'm just going to give the example of each one. What exactly is the co-integrate system? So if you have a TI plasmid disarmed TI plasmid or a binary vector, and then you have your plasmid which you are making in a E. coli, and so, or not a binary plasmid like I'm showing this, that little tiny plasmid. And so what is happening in the co-integrate system, elements from the little plasmid will get integrated into the TI plasmid. Whether it is a binary plasmid or whether it is a TI plasmid, it can function as both, but when research early on, when things were developing, it was the TI plasmid which were integrating these components. This is how, like, TI or binary plasmid or unitary plasmid would look. And then in the agrobacterium, if it is a binary system, again, there will be two plasmids in there, besides along with the chromosomal DNA in it. And so when you are QCing, you have to remember these principles, so that, like, these information, so that you can design your testing strategy or sequencing or QC strategy accordingly. This is the example of ternary vector system, where you have a TI plasmid, there is a binary plasmid separately, which is bringing the trait in, and then there is an accessory plasmid, which is containing a lot of virulence genes in it, so that it can have an enhanced efficiency. This is the technology that was that came from Corteva. So this is the example of that. And one of the things I want to mention is, when we think about the tDNA, we think about one tDNA most of the time for the general people who are utilizing this technique. And you don't have to limit yourself with one tDNA. You could put multiple tDNA in a same binary vector a tDNA could be placed one in a binary vector, one in a TI plasmid, or it can be launched also from the chromosomal DNA. So the key takeaways from my presentation, I would like to say, based on my experience as a facility director and the clients I work with, many times, they are working with their own agrobacterium and delivering to their facility. The biggest request I can have is, or any researcher working on this, know your agrobacterium, very important, because everything you do, it impacts you down the road, several years from now. If you have the issue, it will waste a lot of resources and time. And so it's very important to know what agrobacterium strain you are working, what is the tDNA vector strategy in there, what is the sequence of plasmids and all those things. So this is what, and then do quality check before you start. This is the first thing you should do before you enter in this. With that, I'm going to end my presentation and then take questions for 5 minutes and pass to Kansab Lee.
So because this is being recorded, you have to ask your questions through the microphone, otherwise we have to repeat it. So Prasan has very nicely volunteered to be our runner. So wait for Prasan to come with the microphone. This way people don't have to try to make their way over to the mic. So we have about 5 minutes for questions for Veena.
Hi, really amazing talk to get the basics. Can you please, do you have some examples where the agro strain made a real difference in any system or changing the strains?
Yeah, I'm going to give the real world, like currently what I notice in light of how not many people understand this strain, okay? And so there was a plasmid we were trying to put in the Agrobacterium AGL1. And then nowadays, the vectors, if you think through, they became very complicated and very large with a lot of repeat elements. And so it was not getting very stable, like we were not getting colonies which were, you can count on. And so we have to change this strain, and then it was stable. So it's a rec minus strain helped. And so many times you would start your experiments making your vectors, putting it in the E. coli. Most of the E. coli strains are rec minus. And then you put those plasmids in the agrobacterium, and then what happens is, if it is not rec minus, then there is a potential with a lot of repeat element and a lot of recombinases, CRISPR, and all those things cause recombination, so it would not be stable. But the thing is, unless you QC, you would not know. And so QC and make sure before you do the transformation to see is the plasmid same as what you intended to have. And so this is, what I'm saying, real world example, but you could change the vector in so many ways so that you could enhance the host range. And there are so many things, so we can talk offline on that, but there is so much about this. And then more you read, more you would learn. Thank you for that question, though.
Thank you so much for the presentation. Quick question. You mentioned QC your plasmid. Do you just mean sequencing it, or are there more steps that you take when QCing your plasmid and your strain?
So traditionally, people used to QC the region of the plasmid, PCR and region, but sequencing has become very cheap. And so people may have used Plasmid Source or other companies. And so you should if you could, sequence whole plasmid, because even the restriction digestion could not tell, like, it should ideally, if you are having the same pattern, it may not be enough sometimes. And so if you could sequence, please go for the full sequence. And just don't rely on PCR amplified fragment, because PCR has its own, if there are repeated elements, so it can be challenging to make a real call about the quality. So, okay, thank you again for your attention.
Okay, our next speaker is going to be introduced by Amy Malzahn, who is our plant gene project coordinator.
Like this? Okay. Hi, everyone. Good morning. I'm happy to introduce Dr. Kyansub Lee to you all. Dr. Lee is a research scientist and adjunct assistant professor in the Department of Agronomy at Iowa State University. His research involves the development and improvement of plant biotechnology tools. His group aims to broaden the spectrum of transformable crops by engineering agrobacterium strains and implementing new technologies for genotype flexible transformation. Dr. Lee.
Thank you, Amy, for the introduction. Welcome, everyone. It's great to see you. It's nice weather, actually rainy, so it's cooler. I was sweating a lot yesterday, so. Okay, let me move. So my title is about tDNA vector component. So I'm going over the basics, very basics, about plasmid and the vector design. And then next session, Katie will talk about cloning method. So first of all, I'll give you a very, very brief and general introduction to plasmid vectors. And then I will repeat what Bina already said about unitary, binary, and ternary vector system. And then that can justify why I talk about plasmid. Because when we use a multiple plasmid in the same agrostrain, they must be stably coexist, right? So then compatibility issue becomes very important. So if you don't understand how plasmid work or how construct you need to prepare, then there is some conflict or incompatibility issues that can ruin your entire experiment. So to be consistent, I want to bring this up. This is the glossary for the terminology we use in biotechnology or transformation. And I heard that this was suggested by Wayne Parrott and Piero, that when we use these terminologies, we say the same thing. So construct is an artificial DNA fragment, right? So when we say construct, it is something we have the trait that you want to modify so that we can introduce it into your target cell or tissues so that you can modify the traits. Vector, we all know that vector is something, without vector, construct cannot, replicate within agro, for instance. Or for cloning, we need to put your fragment into the vector so that the final product is, we call it construct. And plasmid is, we all know the plasmid, but I will go over a little bit more detail after this slide. Plasmid is usually circular, autonomous, replicon that exists in many bacteria. They can carry antibiotic resistance genes, but resistance gene, and also have a original replication so that they can replicate without any other restrictions. And then cassette, gene cassette, we say with gene cassette or whatever cassette is also the sequence is usually chimeric. So if we say like a gene cassette, like a reporter cassette, it has a promoter and a coding sequence and terminator, usually flanked by excision enzyme site. But all these days we can use Krelak site, whatever you can mobilize to next level of a vector. Okay, plasmid, usually it's circular. Right, as you can see here, this is a really simplified version. I want to point out three things. One is you need to have original replications. So what it means is that it should be recognized by the host cell, And then the DNA replication machinery can be recruited to initiate replication so that it can be maintained in the cell. Second part is, selectable marker genes. When we introduce them into the host cells, you should be able to establish it, and then that usually requires antibiotic resistance genes. And the third one is cloning, right? You need to have a cloning site. Traditionally, it has been resistant enzyme site. So you digest your vector, and then you PCR amplify or digest your gene of interest or fragment from another vector, and then you can clone it. But these days, Gibson cloning, modular cloning, there are many different cloning technologies developed. So you can use them, but we still call them like a cloning site. So what is your plasmid? I already repeated multiple times, right? So I can skip this one, but I will tell you the history. So plasmid was called plasmid because it was considered as a cytoplasmic inheritant material, right? So that's why combined by 1952, I think the battery is almost gone. Very weak. Yeah, the term was coined in 1952. I just Googled this one. This is not critical part, but if you're interested, you can see how people discover the plasmid, how they name it. Many of the plasmid they carry, they are used for, they are considered as a parasitic or material, and they carry many of the antibiotic resistance genes. That's how bacteria evolve their resistance and they spread. And also they play important role for horizontal gene transfer between microorganisms. But for us, it's a tool, right? We're interested in using them as a tool. This is a basic part. Sorry, I cannot, nothing. It's not connected. It's only connected to their system. Sorry for the inconvenience. So I'm going to say just verbally. So I already mentioned it. There's you need original replication, and then usually antibiotic resistance gene. And there are cloning sites. You can use different cloning method. Multiple cloning sites refer to the resistant enzyme site. And you can have a gateway recombination site or a golden gate modular cloning site. So I brought this up again because this is very important, right? If you are experiencing low tDNA delivery efficiencies, then we recommend a ternary vector system, which carries an extra helper plasmid. So the ternary helper plasmid was developed a long time ago by introducing the constitutively active G as on a third plasmid, in addition to the TI plasmid and your binary vector, that enhances the transformation. But more recently, Corteva developed it based on their knowledge with a super binary vector. So after this slide, I'll go over one by one. But what I'm saying here is, if you want to use a ternary vector system to enhance the tDNA delivery efficiency, you must consider the plasmid compatibility issue. So the binary vector it was developed like 40 years ago, right? And based on the fact that if you put the DNA border sequences, left border, right border, wherever you put it on the chromosome or on a separate plasmid like a binary vector, you can transfer that fragment into the plant cells. So that was the key finding. And then if you put on a separate plasmid, your cloning process is much easier, so easy to manipulate it. Instead of, you clone your gene of interest into the co-integration vector, introduce the agro, and then wait for the recombination, and then you have to check, whether your construct is made or not. It's really tedious, and it takes a long time, and quality check was not also easy at the time, right? But it was much better than original unitary vector system utilizing disarmed TI plasmid. So, but super binary vector became really popular after Japan Tobacco made it, and then they established a very efficient transformation method for monocots, especially corn. Before that, corn transformation was considered almost impossible, but using super binary vector, it was very, very efficient. Again, this vector system has the same issue as a unitary vector system because it requires co-integration step. Therefore, ternary vector system became really popular these days. I hope. At least we are using ternary vector system and we don't have to encounter low transformation issues, at least with the Biono4, for instance. So if you are experiencing the same problem, if DNA delivery is the bottleneck, then this ternary vector system will be very, very useful. So by having a third plasmid on a compatible backbone, so that's what I'm going to cover next slide. So for tDNA, this is a basic part of tDNA. Again, because it's a plasmid vector, it also has original replication. This is used for both E. coli and Agrobacterium. So therefore, usually you need broad host range original replication, which can work both E. coli and Agro, or you can put two original replications, one for E. coli, one for Agrobacterium. This is done because usually broad host range plasmid have a low copy number in E. coli. So during the cloning step, you get only a small amount of DNA. Therefore, by adding high copy number of original replication, you can easily manipulate the DNA. So it's all based on our convenience, practical purpose. So this is a basic part of the tDNA. I want to emphasize one more time. You need to have an original replication and the bacterial selection markers. So you can have only one selection marker, which will work both for E. coli and Agrobacterium. But what's the important part is next, tDNA repeat, right? And then we will have a panel discussion on that, what is the importance, how you can design your tDNA construct. Usually we mean it's your tDNA region. So the tDNA region, I can just say one thing. If you have make a new tDNA vectors, make sure you put your selectable marker gene just before the left border, right? And then traditionally, if you are considering transferring Agrobacterium plasmid between different Agrobacterium strains, original transfer was included. But these days, we are using electroporation, so it works very well. But if your plasmid is really huge, including this original transfer for conjugal transfer is one option. So again, I'm repeating this one more time because it's very important. Original replication, why am I repeating it? If you're putting two plasmid that belong to the same incompatibility group, in other words, if they carry the same original replication, we call it they are not compatible. They cannot coexist in the same cell, even if you put different antibiotic resistance gene onto plasmid. Especially if your plasmid has low copy number, this will be more prominent. You may not get any transformant. If it's a high copy number plasmid, you may get some transformant, but as you subculture them, you will lose one of them. So that's why it's important. So here, original replication refers to the minimum cis-regulated element region that requires to keep the replication of the plasmid. Okay, so we are using the first terminology. And have you heard about plasmid host range? Usually we don't care, right? Because we are getting plasmid from someone who uses it for that, bacterium. But there are different host range in plasmid. Some plasmid have a broad host ranges. So even if they were isolated from Pseudomonas or E. coli, they can be used in many different bacterial species. So here I listed a few of them. So RK2, BBR, and PSA. These are really broad host range original replications. they can work for both E. coli and agro. And that's why the early version of binary vectors was based on the RK2 because it's broad host range origin replication. It's called a shuttle vector. It can move between E. coli and agro. So the next one is original replication for E. coli. These are really commonly used, but you will be surprised they are actually very similar. They are derived from the same plasmid. coli1, PMB1, PBL322. They only have a few base changes that made the copy number changes dramatically. So this will be true. So there's a area we can explore. If your binary vector you suffer low transformation frequency, you can make it to have a higher copy number. So if you have a high copy number, there's a chance that you produce more tDNA within agro cell. Therefore, you can deliver more cells to the plant. The bottom one is original replication that only work for agro, that do not work for E. coli, which is PVS1 or root-inducing plasmid original replication, like a PRI. If you only have only this one for cloning, it will not work in E. coli. So you have to make sure that you include another original replication just for the one in the middle. So these days, most binary vectors, they combine one for E. coli and one for agro, because you can get a lot of DNA during cloning, so therefore cloning process is easier. But in the end, final product is the same thing. So here are the I'm listing 4 groups of binary vectors, original replications used in the binary vectors, meaning that you can mix and match. Group 1, I grouped the 4 of them. You can mix and match with anything from different groups. So for instance, the first one is incompatible with P-1, like RK2. You can combine with any of these, right? So if we are thinking of making, let's say, multiple tDNA vectors, Veena said multiple tDNA on one binary vector. Another option is you can put 2 binary vectors, or each one carrying different tDNA. As long as they're compatible, you can put them into the same cell. So I'll tell briefly about what is the mechanism of incompatibility. So incompatibility means that when we put more than one, let's say 2 different plasmid, here color-coded in blue and pink, if they are stable, how many times you subculture them, they should stably coexist. However, if they are not compatible, you will lose one of them. So after some subcultures, you're losing one of them. One of the very well-known mechanisms, we don't have to go too much detail, but if you're interested in the plasmid incompatibility, this is one of the well-known mechanism. Again, RK2 plasmid backbone. It has been studied, and it has a iteron region, which is repeat region where the replication initiator protein bind, right? At low copy number, which meaning if the plasmid copy number did not reach the maximum copy number per cell, the replication initiator protein will bind to the regulatory region, will activate the DNA replication. However, if you already reach the maximum copy number of the plasmid, meaning that you will have a lot of initiator proteins, they form dimer, and then the dimer forms will block replication because it's called a hand curve. When you reach the certain copy number, the initiator protein will block the regulatory region so that it can prevent the DNA replications. This is one of the known mechanisms. Another mechanism is even if their replication machinery has no conflict, but if their partitioning mechanism is not compatible, meaning that the cell machinery do not recognize them as a separate and compatible plasmid, what happens is that during partitioning, there's no guarantee there's an equal distribution between 2 daughter cells during cell division. So therefore, especially low copy number plasmid, the chance is very high. You will lose one of them very quickly. So to summarize it, I went over different vector system, and then I briefly summarize it. tDNA binary vector component. You need to have border sequences, cloning site, and antibiotic resistance genes. And then we'll have, I think, a panel discussion on tDNA region, because that's the most important part. But I was just covering different vector system and plasmid. So I told about unitary and binary and ternary vector system. If you are experiencing low DNA delivery efficiency, ternary vector system can really boost your transformation. And to have a compatible plasmid, if you're thinking of using ternary vector or using multiple tDNA vectors, you must consider what are the compatible plasmid. You need to have a compatible original replication, meaning that you cannot put two plasmid with the same original replication, at least. You choose the one that belong to different incompatibility group. Second, even if you do that, you must have a different antibiotic resistance genes. You cannot use the same antibiotic resistance gene for different plasmid. Third consideration is agrobacterium. During the development of agrobacterium strains, they carry, some agro strain carry different antibiotic resistance gene in the chromosome or on the TI plasmids. So therefore, you have to check existing antibiotic resistance in the strain that you can select, what are the available antibiotic resistance genes you can use for your binary vectors. Okay. So this is the considerations of tDNA vectors. I already repeated multiple times. Agrostrain, as Veena said, we know agrobacterium is a pathogen, therefore it does have a host range. Some strains work better for certain plant species, and therefore you can explore them. Another thing is their genotypes, right? The Reg A minus strain, such as AGL1, it can support a larger plasmid because it will suppress the recombination between agro cells. If you're considering ternary vector system, again, think one more time about the vector compatibility. And then plasmid copy number is another thing. So let's say if you are experiencing low transmission frequency, your goal is delivering as many tDNA as possible, right? And using ternary vector, it's good. You can also consider using a high copy number of a binary vector. So that will increase the number of tDNA, T-strand production in agro. So extreme comparison was that you insert the tDNA into the chromosome, right? You will have only a single copy of a tDNA region. You can have a high copy number plasmid, up to like 10 copies per cell. Then if you compare the transmission frequency, you can see there's huge difference. However, there's some trade-off. If your DNA delivery has no issues, but if you are more focusing on quality event, meaning that's clean single copy delivery, then low copy number binary vector can be useful for that. Okay, so for those who are interested in using ternary vector system, these are available ternary helper plasmids from Adagene, so you can request them. A couple of them we deposited. So the newer one will be available soon. So the manuscript is under revision, so will be available very soon. With that, thank you so much for your attention, and I'm happy to take any questions.
From me.
You can use it.
I hope. Okay, yeah. Katsab mentioned, so one agrobacterium strain can have multiple plasmids. And he also mentioned you should be careful and then have different antibiotic resistance on those plasmids. And so if you have that agrobacterium Having these components ensures that plasmid is compatible, but make sure your media have those selection agent. Otherwise, you will lose it again. And so because it has there, it doesn't mean it is going to survive or stay for long if you don't keep on selecting. So one of the things,
Yep.
so don't forget that.
Thank you, Vina.
Thank you for the presentation, Ken Sub. I was just curious, what's the main difference between the binary and the super binary vectors?
Yeah, thank you for the question, Connor. The binary vector, we all know that. If you put both the sequences on a self-replicating plasmid, that's called a binary vector. Super binary vector is called because it carries extra virulence genes. It was called a Komari fragment. It comes from a super hypervirulent TPI BO542 plasmid, and then it contains the VRG and the VRB and VRC sequence.
Okay, thank you.
So it's nice to have the turn resistant to add extra copy of the virus genes, but should we consider the fitting cost of the plasmid? Fitting cost, because you're adding more plasmid, actually the bacteria may have the fitting cost.
Yes, so she was asking if there's a cost for having extra plasmid. Yes, that is true. You know, if you add additional plasmid, you need to add antibiotics, right? You are increasing full dose of antibiotics that agro will be suffering, right? So therefore, if, in case you are using multiple plasmid, then you can lower the antibiotic concentration, for instance. So for instance, if you're using single antibiotics, spectramycin, we usually use 100 milligram per liter. When we use multiple, we cut it down to half, about 50 milligram. But during the transformation, we use a full dose. But for maintenance, you can reduce the concentration.
Hello. So my question is a little bit more broad. So can you comment on trans-tDNA suppression?
tDNA suppression?
Trans-tDNA suppression, where you have multiple similar sequenced tDNA fragments in one background.
In the same cells?
Yeah.
In the plant cells,
Yeah.
you mean?
Like, so we talk about tDNA transformations, introducing tDNA fragments into the plants, but I guess because I'm having some of that problem with my project.
Yeah, I think we can do that during panel discussion. That's a really great topic. Thank you.
Okay, now, as I mentioned earlier, this workshop was based on feedback that we heard from the community, especially students. You wanted to hear about, how do I put my vector, my construct together? Where do I start? So for our next speaker, we decided to engage with the next generation of plant biotechnologists, and we invited Katie Toomey to give a presentation. So Katie is a second-year PhD student in Wayne Parrott's lab at the University of Georgia. She works on genome editing of poplar and muscadine. She also, in her downtime, she likes to knit, garden, very close to my heart, Katie, and pursue side projects in the lab. And I can tell you, she's also very artistic, not only in the lab, but in her drawing. So Katie is going to tell you about the basics of cloning.
Okay. Thank you all so much for being here. I know I'm the last presenter, so hopefully y'all are still bright-eyed and bushy-tailed because there's a lot more to do today. I'm so excited to be here. I hope y'all all get a chance to go see the Arch. I can't believe how close we are. I'm still young and traveling is new to me, so I'm really excited to have this opportunity. I'm just so excited to be here and talk to y'all. So, thank you for that wonderful introduction, Dr. Joyce. And like she said, I'm here to give you basics to cloning and vector construction. Now, there is so much more than what I can tell you in 30 minutes. And for that reason, I made a little website where I essentially brain dumped everything I'm going to talk about today. And there's also all the notes that you would need as well. You're welcome to take notes as well. You can take as many pictures of my presentation as you'd like, but everything you'll need, including various links and resources, are also available online to some protocols and where you can also purchase some reagents. Non-sponsored. So, this was already kind of established by Dr. Vina and Dr. Kinsub, but I'm also going to reiterate it just a little bit. So, there's some common jargon that's used, and I'm going to explain what those terms are for the sake of my presentation. Sometimes there's some nuances, and people may use these terms a little interchangeably, but for my sake, a plasmid is an extrachromosomal circularized DNA that is able to be uptaken by your cells. A vector is a plasmid that is able to take up an insert. And here is an uncut and cut vector. What I will call a genetic element is what is referred to as a promoter, coding sequence, terminator, essentially just a component of your cassette or insert. Cassette is specific to having recombinational elements on the sides, whereas insert is what's known as a transcriptional unit. It is all of your genetic elements combined. And your vector plus your insert is what's called your construct. Now, the process of getting your construct is cloning. So, now that you know what it is, how can you use it in your research? So, like Dr. Joyce said, I am a little artsy. Here's some chromoproteins that a coworker of mine actually cloned, and when he had so many of them, I asked him if I could paint with them. So this is an LB plate that I made a little rose out of chromoproteins. Now, you can use cloning for so many more useful things, but I just wanted to show the art side behind it. So, you can also use this for products. Maybe you've heard of the Firefly petunia or the Syngenta or Norfolk tomato, which I believe was even at the last SIVB. So you can use it to transfer whole cassettes or transcriptional pathways, but you can also use it to do gene editing so that you can knock out genes and study their function like that or overexpress them. You can also use cloning in ways that I haven't done, where you can use bacteria as factories to essentially create proteins like insulin, et cetera, There's so many things you can do with cloning, but I'm going to focus on the plant side of it because this is plant gene. So, I'm a little bit different than the other presenters in that I'm going to give you a broad overview of how you go from pen to paper, how you go from your computer to your plant. And for that, there's 5 modules. In silico, in other words, on your computer. Molecular, in other words, at your lab bench. E. coli, which is still at your lab bench, but also mostly in a plate. Agrobacterium, which you've heard plenty about today. And lastly, going into your plant. So, in terms of in silico work, there's 2 sub-modules for that, which is your setup and your assembly. I included the QR code again, just in case anyone missed it and wanted to catch up now. I see some phones, so I'm going to give it a minute. Okay. So, you want to ask yourself the big life questions. What do you want to do? What do you want your construct to do when you move it into your plant? What will your control be? Because I can tell you not a lot of people think about that when they transform. And do you need a full-length gene or do you need just a partial? What kind of terminator do you need? What kind of promoter do you need? We're going to get into all of that. But I just want to remind everyone to think about this really early in advance and talk to your PI about it. So, the general components that you're going to need, and y'all are all familiar with these, are the promoter, the coding sequence, the terminator, and potentially your signal peptides. So, to go into a little bit more detail, let's talk about that promoter. Now, there are promoters that are specific to your monocots, specific to your dicots, and there are ones that can be used for both. There's also constitutive, in other words, always expressed in all cells kinds of promoters, and there's also cell-specific promoters or inducible promoters. So in my lab, we use what's called a heat shock promoter. In other words, it'll only be induced at a certain temperature. There's also cell site-specific markers, so that maybe you don't want your leaves to be expressing a marker, but you only want the fruit to express it. If you can find a promoter specific for that, you can use it. You also want to consider your signal peptides. Now, the most common of these is the nuclear localization signal, which is used for genome editing because that's where the nucleus is. But there's also other organelle-specific signal peptides that you can use, such as endoplasmic reticulum and chloroplast signal peptides. It can be upstream or downstream of your coding sequence, or both. And then there's the coding sequence, which is really specific to your project. There's not a lot to say here, but I'm going to say don't forget those stop and start codons. Those are important. And also, if you're having trouble with it being expressed in bacteria, consider looking at the introns. And if you're trying to express it in bacteria, get rid of the introns. And lastly, there's the terminator. Again, not too much to say here, but here's some commonly used terminators. We use Rubisco terminator, and there's also the NOS terminator and heat shock protein terminator. So, all of these elements together create your insert, in other words, your cassette, and once you have a linearized vector, they can go into your vector. And now I'm going to talk a little bit about the in silico assembly. So, once you have all of your components identified, you are going to need to stitch them together on your computer before you start putting them together in the lab bench. So you need to visualize this and make sure that it's all going to work out. There's some software that's available to you. I recommend Genius Prime. This is a really great resource if you can afford it. It is really wonderful. It's really user-friendly. You can do restriction enzyme digest through the software. You can also do type 2 cloning through the software. Really highly recommend. But if you're a graduate student or an undergrad, you may need the free software, which there's also one called APE, a plasmid editor. And this is a little bit less user-friendly, but it's still really, it has all the components you need to get started. You can still find your restriction enzyme sites and it'll highlight them for you. You just have to do a little bit more manual work. I've used both, and they're both fine. So, once you have your construct completed, there's also the backbone that I don't want y'all to forget about, so I'm going to gray out the insert for now. And I want to say that there's a plant marker there, which can be antibiotic or visual, and there's also the bacterial marker, which is usually antibiotics, can be visual. And we're going to go into this more later. I just don't want y'all to forget that those are there. So, let's go into the molecular side. You're finally done with your assembly online, and you're ready to get started. So, you have to obtain your insert, build your construct, and transfer it to E. coli. So, there are various ways to obtain your insert. If you're so lucky as that your plasmid is something that's already been made, you can go to plasmid repositories like Addgene and purchase your plasmid there. Now, Dr. Amy has compiled a list of plasmid repositories that are available on my website and also through the PlantGene website, I believe. And so there's about 8 or 9 different resources there of full plasmids. Another one is Arabidopsis Biological Resource Center. They're really good for having tons of plasmids specific to Arabidopsis. And both of these have complete plasmids, complete constructs, but you can also use fragment synthesis from companies such as Twist Bioscience, where you can just order the insert that you want and use it from there. Now, these cost money. If you can't afford that, then worry not. We're going to go into molecular techniques to obtain your insert at the bench. Now, the general premise behind this is you have your insert and your vector. You want to make them one. So you have these overlapping sequences that are going to be denoted in orange and green throughout my presentation. And you have to add them to your insert somehow and create sticky ends so that it can be actually inserted. Oh, yeah, I'm highlighting the sticky ends there. So the 4 techniques that I'm going to go over are restriction enzymes, PCR cloning, ligation-independent cloning, and Gibson cloning, which is also known as any builder. So there are so many more techniques out there, and I listed a couple on the website. These are the 4 that I think people should be familiar with, especially when you're starting to clone. If I had to recommend just one, it would be any builder, but I'm going to talk about all of these because I think it's important that you understand the basics. So, restriction enzymes create short overhangs. Hopefully everyone's a little bit familiar with what those are, but if not, let's say that you have a restriction enzyme site like EchoR1 and HinD3. You want to make sure to have 2 different enzymes because if you have one, you risk your insert being flipped. So this allows for unidirectional insertion. And after it digests both your plasmid and the insert, you have acquired those sticky ends on your vector and your insert. At this point, it's just simple ligation to build your construct and then transfer. So that's restriction enzymes, potentially the simplest one. Now we're going to get into PCR cloning where it gets a little bit more complex. Everyone's familiar with PCR itself, but what was new to me when I joined graduate school is that you can actually add to your amplicon with your primers. I'm sure that some of y'all are just like, well, duh, but this was interesting to me. So that works like this. In your thermocycler, you essentially have your overhangs that are amplified through the PCR. Now, to use this in cloning, there's 2 general methods. You can either do mix or incomplete PCRs. Mix is essentially where you have 2 different PCRs going on with overhangs being amplified in each different reaction. And then with both your products, you boil to separate and anneal to combine. And a portion of your products will have the required overhangs, the required sticky ends. Then you can also do incomplete PCR, which is essentially where you terminate that final extension step. So instead of a completely amplified amplicon, you get a partially complete. So with both of these, you have acquired your sticky ends, and you move to ligation. With the case of the incomplete PCR, you would also need a polymerase step because it's not complete, and you may need to fill in those gaps. But moving on to one that doesn't require ligation is ligation-independent cloning. So this does PCR, similar to the PCR cloning, where you add your overlaps, but you're using a unique polymerase called T4 that will actually act as an endonuclease or exonuclease as well. So you have to have a limiting nucleotide in your overhang, and you have to amplify your insert and vector separately. So let's take a closer look of what that would look like in this orange overlap. So as you can see, in the insert, there's no G, and in the plasmid, there's no C in the orange overhangs. Here's the first ones. So the exonuclease would chew back up until this point, and then simply anneal from that point on. Now, if you've fallen asleep, I suggest you wake up because it's time for Gibson/any builder. I like to refer to this when I'm talking to my undergrads as the 3-2-1 reaction because it has 3 enzymes. You can use 2 or more fragments, and it's all done in one tube. Now, I want to say you can actually, when I say 2 fragments, I'm talking about the insert and the vector. You can use this for what's known as multi-cloning, which is where you have multiple inserts, but this is a little bit too complicated for what I've been told to go over for today. So your three enzymes are your exonuclease, which is in this case is T5, not T4, so it's not nucleotide dependent. And it's going to chew back those ends and create those sticky ends. But because it's not nucleotide dependent, it's not going to have a specific stopping point. So when you ligate them together with your ligase, there's going to be some gaps. And you're going to use polymerase to fill in those gaps. So that's the end of Gibson. That one's really simple, and I really recommend that. And I think it's the one that we use most in our lab. So to give a brief summary to those molecular techniques, the ways to obtain your sticky ends are either PCR cloning, where you make your overlaps, you purify them, anneal them, and combine with your insert or your plasmid. There's also ligation-independent cloning and restriction enzyme, which both create those sticky ends through chewing back, and then you combine with your vector. And Gibson cloning, which all happens at once, where it creates your sticky ends and combines them in one reaction. Now, you have your plasmid at this point. It's in a tube. You cannot see it. You don't know how much is in there, but you want to make more. So you move it to these little factories called E. coli. And luckily, you have there are what's known as competent cells. In other words, they are prepared beforehand to be able to take up plasmids. So there's two that I recommend for beginners called DH5 and 10-beta. 10-beta is good for any plasmids that are 10 kb or more. 5-alpha is for smaller than 10 kb. They're both available through NEB. However, you can find them at many other resources as well. This is just what I use. And the way that it works is rather simple, that you take your reaction that you hope your plasmid is in there, put it in your tube with your competent cells, ice for about 20 minutes, heat shock for 30 seconds, and ice for another 5 minutes before adding your nutrient-rich outgrowth media. And depending on your antibiotic, you can either plate right away or put it in the incubator for a little bit. And then it just goes on your plate, and you hope to see colonies in the morning after you incubate it overnight. So with that step, we're going to move on to the third module, the E. coli step. So you don't know which cells have your plasmid and which don't, which is why you're going to do the selection, screening, before you transfer it to agro. So selection is the passive process of selecting your E. coli. This is something that your plate will actually do for you if you use the correct antibiotics. And you can either do positive or negative selection. Positive is where you're looking for a gain of antibiotic resistance. So an example of this is you have however many cells you have, and you know that some of them should have your plasmid, and that plasmid would have your bacterial resistance marker, let's say for kanamycin. So you put it on a kanamycin plate, and the cells without your marker will not be able to grow. There's also negative selection, which is where you're looking for the loss of a trait like CCDB, so that only those with the recombinant will grow. Now this differs from screening, which is where you as the human are the active filter in selection. So a common example of this is visual screening with blue and white screening. This is a little bit of an old school technique. There's a plate right here, so you essentially can see which of the colonies have been transformed by interrupting a subunit for the blue protein. So essentially your white colonies are your recombinant. This is a little old school. I think only one person in our lab uses it, but more commonly what we do is PCR screening, which is really easy because you can screen your colonies before you grow them up in liquid broth. So you essentially just have your PCR. I'm sorry, I see some of y'all are laughing at me calling it old school. It's still great, it still works. So you do your PCR in advance, and you make sure to have a primer inside your construct and outside, because you don't want to just be amplifying the DNA that you put on the plate. You want to be amplifying those colonies that are growing. So this will be able to tell you if your colony is positive or negative before you grow it up, which is really helpful. But it's also really helpful to have more definitive results, which is why after you grow it up in liquid broth, you mini prep it, which is essentially where you extract the DNA from the plasmid. I have protocols for this on my website, but I'm not going to go into it here. And you can do restriction enzyme screening, which will give you more telling results. You essentially look at your plasmid in silico on your computer, see where your restriction enzyme sites are, make sure that when you digest it in real life and you run it on a gel, it matches the in silico results. And there are examples and little quizzes and stuff on the website for fun. Now, after you've done your selection and your screening, you may think you're done, and you can be, but I would highly recommend sequencing it if you can. Pretty much anywhere that does Sanger sequencing will do this, but I recommend Plasmid Saurus for whole plasmid sequencing. It's about $15 plus shipping and handling, and you get your results in about 1 day, 1 to 2 days. Honestly, they've given me my results on a Saturday at like 3 p.m., so praise be Plasmid Saurus. There's also a Zenta, which is really great for if you're just looking to amplify your insert. It'll be about 800 base pairs for $5, and plus shipping and handling, and you'll get your results in about 2 to 3 days. But like I said, anywhere that does Sanger sequencing of plasmids will be great for this. So I'm going to take a sip of water. After you've confirmed your sequence is correct and you're all confident about your construct, it's time to move it to agrobacterium. Now, you've heard a lot about agrobacterium today, so I'm just going to go over the simple confirmation once it's in agrobacterium before you transfer it to the plant. So the way that we transform agrobacterium in our lab is through electroporation, which is essentially sending an electrical current through your agro to electroporate holes in the cell membrane that'll allow it to take up the plasmid. And then you grow it on your selected media so that you can select for only those that have taken up your plasmid. Also, I know that this is not an accurate agrobacterium. Avena's was better, but please forgive me, this is just cute. And you can also do PCR amplification to confirm that your plasmid is there. This depends on your risk tolerance. I know some people that, like myself, who amplify from LB to RB, your left border to right border, your whole tDNA. That's what I recommend if you can. I know some people that just select for the coding sequence, and I know some people that have the whole plasmid sequenced. It's all dependent on your risk tolerance and your budget. So I'm not going to go into plant transformation protocols because there are so many more crops than what I can go over today, but I'm going to give you some general considerations for your plant transformation. I'm going to start with the backbone because you have your plant markers in your backbone, which can either be visual or antibiotic. And with your visual, you can have what's called colorimetric, like Ruby, if you've ever heard of it, where you don't need a fluorescent system. It'll just change the color of the cells. And you can also use fluorescence proteins, but that requires the flashlight system, like I said, where you have to fluoresce your cells. And that flashlight system can be expensive depending on your budget. You also want to make sure that there's no autofluorescence in your cells. So if you're checking for a red reporter, like RFP, be sure to look at your cells in advance to see that it doesn't look red already, because that has happened to me. And with your antibiotic marker, you want to make sure that your antibiotic has worked in your system before, because I know kanamycin has some problems with some monocot systems, and there's also some just limitations of using all antibiotics in all plants. And you also have to consider, does your antibiotic need light? Because some, like spectinomycin, they use the chloroplast for their selections, so you have to make sure that your plants are actually using their chloroplasts in the light. And if you're doing a transformation system in the dark, this is not going to be helpful. So, as an example, let's say that you have kanamycin as your plant selectable marker, and you also have your bacterial selectable marker that you can't forget about. So I'm going to denote the antibiotic marker for plant in blue, and the bacterial resistance in orange, let's say hygromycin. Now you can't forget that you also have put all of this in agrobacterium, so it will have its own resistances as well. Vina mentioned agrostrain GV3101, so I'm going to talk about that. It is resistant to gentamicin and rifampicin, and so when you're working with anything with the agro itself, you should use gentamicin, rifamycin, and hyromycin. And then after your co-cultivation period, you'll use something like meropenem to kill off your agrobacterium so that you only have kanamycin marker, which has been transferred into your plant genome. So to give a brief overview of what that looks like in your timeline, and this is a little bit malleable depending on your plant transformation system, but essentially when you're working with anything bacterial, you're gonna have your bacteria resistance. So your hygromycin in this example, but whenever you're working with agrobacterium, you're gonna have your agrobacterium, gentamicin, and rifampicin. And then your co-cultivation period before you use meropenem and your plant resistance to select for your transformed cells. So with that, I'm going to end my presentation. There's plenty of resources that I've listed here that I recommend you looking into if you're just starting to clone, and there's also plenty more on the website, and I just wanted to thank you all for being here. And I am taking questions.
Thank you for the nice presentation. When you use the same selective marker for like a kanamycin resistant for plant and a bacteria, if you having different promoter, can like is it okay to put the same selection instead? Do you have a recommendation for the if avoid kind of leaky expression of that type of genes?
So the promoters will be different because the bacterial promoter will be specific to bacterial and plant for plant, but also for kanamycin, like you said specifically, have different coding sequences for the plant and the bacteria, so there shouldn't be any overlap in that case. But if you're worried about it, maybe check the literature and make sure that there's no leaking of your plant promoter in the bacteria or vice versa.
Okay, let's thank Katie again for that great step-by-step. So just a reminder, this is, workshop is being recorded. So in addition to Katie's great resource that you can get that she put together, we'll also have the recording first on the SIVB site, and then once it's released from its contract, it will be on PlantGene for as long as PlantGene exists. So you'll be able to access that information. All right. So what we're going to move on to next are the helpful hints presentation. Thank you. All right. So this is the interactive part. So we are going to solicit questions and also input from all of you. And also this is time if you've thought of a question, since our speakers gave their presentation, you have that opportunity here as well. So yeah, I think what makes it a much better workshop is to have input. So please feel comfortable to make Prasan run around and ask questions and give input. All right. So what we're calling this is tDNA design and cloning tips from, well, actually, it's also pitfalls we learned the hard way from the, I know it's suspenseful because I have to, PlantGene Steering Committee. And as I've like to say, is we have 170 years collective experience. So we are great, some of us more than others, but we have a great resource for you. So I just wanted to, I don't have the pointer, but from left to right, Amy Malzahn, who's from BTI, and she is our project coordinator. This is in the front. Next is Vina, who you all met. Heidi Kepler, who is actually one of her children is getting married this weekend. So Heidi is from the University of Wisconsin. She wasn't able to join us. Bill Gordon-Cam, who you saw earlier. Nigel Taylor is from the Danforth Center, and he will be joining us. Next in line is Wayne Parrott. Unfortunately, Wayne, well, not too bad for him, he's at a board meeting, but he wasn't able to be here. And then you heard from Kunsub Lee. And again, if you're not a member of PlantGene, we have that QR code up and become a member. It's a great resource. We have about 860 members worldwide now, and it's really exciting that we've done that in roughly 2 years. Okay. So what we did was we went through and decided that, what have we learned, what would be helpful to you, and what we're going to do is bring up these topics. Members of this group that are sitting here will comment, but we'd also like to hear about your experience as well. So we've arranged this in different topics, and one of those is the arrangement of vector components, which you could learn the hard way, right, is really important and sometimes is not very much appreciated. So one of those is consideration is the arrangement of head-to-head, tail-to-tail, or head-to-tail can affect expression. Surprise? It can, and it's something that you really need to take into consideration. Is there anybody up here that wants to comment on that arrangement? No? Okay. Anybody in the audience want to share your experience with that or putting it together? No? Okay. Well, over there.
Yeah, this was actually on my list of questions because I had a cassette where the terminator of one cassette was right next to the promoter of the next, and I don't think I got any expression of the one that the promoter was right next to the terminator. Is that something other people have seen before?
Anybody else seen that experience? No?
So
You know?
I can start off by commenting on a couple.
On.
Yeah. By commenting on a couple experiences that relate to that. We use Wishel and Baby Boom to promote transformation in cereal crops. And we have, over the years, we have made hundreds and hundreds of constructs with Wishel and Baby Boom. And when we were starting off in this process, gene order was our first important consideration. And what we found was that, so for most of our transformation methods, we want a low level of Wishel expression, so we use a NOS promoter driving Wishel. We want high expression of Baby Boom, so we use a strong ubiquitin promoter. And we found that if we have the ubiquitin Baby Boom upstream of the NOS Wishel, there's absolutely no expression of Wishel and the transformations. And so our speculation, and we didn't validate this, but I think this is probably what's happening, is that the terminator for the Baby Boom cassette, terminators aren't 100%, and so you get some read-through, and transcriptional read-through can downregulate the second gene. So that's one thing to keep in mind. But also so I mentioned terminators reduce read-through. Ideally, they would be 100% effective and stop everything. But different terminators with the same promoter can really impact your efficiency and your overall expression. So some terminators don't work as well in corn, for example. In our experience, the NOS terminator isn't as effective as some terminators that you would clone right out of maize. So just a couple points that might help with that.
Oh, one thing I want to add in there is as, we are talking about different cassettes with different terminators, and terminator is not a stop sign, perfect stop sign. And then if Bill and I received a vector from Bill, I made my own construct, derivative of that with my own gene cassette and additional cassettes in there. Then I also think about, if selection agent worked for Bill's plant species of interest, and then I am doing my own work, and maybe I'm working on the same plant species, selection agent in my media may not work that effectively. So what Bill had success, maybe I will not have success same way. And so think about those things when you're putting your constructs together, because you think about all the time vector, but also how it impacts your media strategy. I believe next workshop where we are talking about the media and all those things. So it's very important, in context with the whole experiment.
Great. All right, I think we'll move on to the next topic, which is there's polarity to tDNA transfer. And you heard a bit about tDNA from both Vina and Kunsub. And it's important to place your selectable marker before the left border. I've seen people make the mistake and not do that. The left border, keep in mind, is a slow down. It's not necessarily a stop. You can get read-through. You can get part of that into your plant cells. And truncations at the left border are common. And kind of how I remember it is the left lags, so that when I was putting my constructs together, I would keep that in mind about the left border and right border. I think there's sometimes a lack of appreciation for that tDNA transfer, and that's why we started with the two presentations to give you the basics that will help you avoid some mistakes, some of the pitfalls that we've learned over the years. Anybody want to expand on that comment about the left border? Anybody else? Questions? There's a question over there.
I'm getting my 10,000 steps today.
Good. Good. You could have a big lunch.
Hi, this is a great idea. I seem to remember some older vectors often had overdrive sequences at the maybe left or right borders, and I often don't have the bandwidth to sequence my integrated tDNAs. So how much of an issue is backbone integration and strategies for getting that left border and preventing integrating too much backbone in my products? So I'm wondering if you could comment on that.
It's a big problem, especially if you're thinking about quality events. And then if you don't have a quality events, you would not be able to assess the trait you are trying to figure out, because there will be implication of those backbone in there. So there are strategies in place besides the border sequences. You could put additional cassettes in the backbone to reduce the survivability of the plants which contains the backbone. So quality events are really still a low percentage, and it's a holy grail. We all try to figure out what we can do to enhance the efficiency. And I have a request. Anybody asking question, please introduce yourself and where you are from so that we can learn about each other also while you are asking question. Any comment, Bill or somebody?
So I want to add a few basic information is that there's a VOD2 protein from Agrobacterium. When it processes the T strand from within the cell, it attaches itself to the right border. It cuts a 3 base from the left side and then attaches itself to it. Therefore, right border region is more protected. So during the tDNA integration, left border side is naked, only protected by the VOB2 protein within the plant cells. So therefore, you can get more truncation in general. By adding double left border is known to be enhancing the processing there. So, however, you cannot completely avoid integrating backbone sequence or sometimes agrobacterium chromosomal sequence because of BRD2 protein itself. It will attach to the single-stranded DNA. If there's a single-stranded DNA, it may attach to itself, can lead to the plant cell. So by nature, we cannot completely avoid this problem, but by adding extra left border, double left border, triple left border, something you can imagine and you can test it out.
And just a quick follow-up, are there any downsides to having a multiple left border that you know of?
What you'll find is sometimes there's a big knowledge gap in information, and it's the sort of thing where it takes funding and time to do those comparisons, but it's the sort of thing that would be really helpful to know, right, as well.
Well, we We have used triple left borders quite a lot. We did quite a lot of work to try to minimize it's not just vector backbone integration, but going right through and getting a tandem repeat as well, and other weird things. So people have asked this question, does it work? Is it helpful? I think it does work, and it helps a little bit. It doesn't solve the problem completely, but we have integrated triple left border into pretty much all of our product quality gene constructs now. It's something we use pretty much all the time. So that I don't think there's really no downside to it. And our regulatory people have looked at this as well, and they don't have any issue with it.
So if you're really worried about this problem, there are also examples in the literature where people have taken a negative selectable marker and placed it outside the left border. So if you do get that read through and you've integrated sequences beyond the left border, you can select against those. It's not always 100% effective because sometimes you'll just get a piece of that gene and then your negative selection isn't effective, but it's an option that's out there.
Can I comment on that as well? We have done that, looked at that as well. But then we found it's like, well, if we didn't, we still ended up having to pretty much sequence or check everything that we got. It does help to eliminate if you're getting a lot of rubbish or stuff you don't want, but we ended up thinking, well, we still have to go in and check. So we didn't go forward with that approach. But it could certainly work for some people, and it also one of these things that helps, doesn't solve the problem completely.
One comment there, like if you have 3 left borders, it means you have 3 repeats. And so remember, there is always give and take. What is the purpose? And then if something don't work, then that is something to think about, especially because nowadays the vectors contain recombinases, which impacts. And so it is, if something works, it's all good. If it doesn't, then think about what you have put in. So know your vector, know your agrobacterium.
Any other questions or comments on that topic? No? Just wanted to reiterate, Vina had a good idea. When you ask a question, give your name and your affiliation, and that kind of helps build our community. And I'm going to lump the next two together. There's the orientation of cassettes and promoter elements in your tDNA that need to be considered. And we talked a bit about the location of selectable marker cassette in tDNA. But if anybody would like to talk a bit more about that, I think it's pretty straightforward. Any questions? Okay. All right. So let's move on to the next. So promoter information. Promoter-terminator combinations can affect gene expression. I think we often think maybe that the terminator is kind of a benign, not important part of, and we'll talk a bit more about terminators in the next slide. But this is something that you need to consider, especially if something's not working for you, is what are your promoter combinations and your terminator affecting expression? And anybody want to add to that, what you might have seen or some recommendations on how to determine this?
Somebody from audience would like to say something?
No? Okay. All right, well, just something to be aware of. And then we all know about 35S, but there are many versions of 35S. And one thing that I think has become, we came up with our steering committee members, is that was also something new to me that I learned from our committee member, was that 35S can work in trans on other vector components. Did you guys know that? That it can work in trans? Maybe the person who told us that can expand on that a bit? Or was that Wayne, who's not here?
I'm no expert on this, but I mean, it's well known.
Mm-hmm.
So you have to be careful on that. If you're using in the same tDNA, if you're using, say, if you're trying to get tissue-specific or expression, if you have 35S driving your selectable marker, it could be interacting with your gene of interest, and therefore undoing a lot of your tissue-specific expression that you're looking for. So it's just something to be aware of and to be careful of. But I like that as be aware, not watch out, be conscious of these things and think about it when you're using them.
One more thing I would like to add here is, that you're taking the tDNA and intention is to bring this tDNA into the plant cell and insert into plant genome. And so it is going to insert within the chromosome somewhere, which could be next to a promoter. There could be a position effect, and then the euchromatic or heterochromatic nature of the chromosome would impact the expression. So if Kansab got high expression or he published things, it is always high expression. And then I am analyzing my event and it's 35S and I say, oh my gosh, what did he give me? Or it is not working. And in academia, many times we don't care so much about the junction where it got inserted. And so it's very important, like in industry, it's very benchmark to figure out where the insertion happened, but those things also matter. So what you put in is important, that's what we emphasize today, but where it got in is equally important. So remember that also.
Katie?
And if I can add something, yes, that does sound like something that Wayne would add, and he's my PI, and if I could I can hear him in my head, and what he would say if he was here is viral promoters have lots of enhancers, but they're not inherently only seen in viral promoters. They're also in plant promoters as well. So just be sure to match the promoter's strength to your level of expression desired, not necessarily always going with viral promoter being directly seen as something negative, that it can have the self-silencing and whatnot. That is seen with 35S, but it can be seen with enhancer-heavy plant promoters as well. So don't just assume that because it's a plant promoter, you're not going to see that.
Yeah, one more comment to 35S that we're aware of is if you're using an excision system like Crelox, watch out by having 35S in there. Again, if it's working in transit, it may be stimulating the excision to happen really quickly in an uncontrolled manner. So that's another piece of it. We're using an excision. We're staying away from 35S for the selectable marker or any other components that we want to be in there.
So this gets, as we discuss this, the tDNAs are getting more complicated and more nuanced by the minute. But so that's a really important point. If you have a promoter that you want it to be off at certain times and then you want it to turn on when you want to do something like excision, 35S can cause premature excision in those types of systems. But 35S, there's also a distance component. If you move 35S away from your heat shock promoter, for example, down to the other end of the tDNA, you can mitigate that enhancement to some extent. And people also have done a lot of work trying to find enhancer elements that'll block the influence of 35S. Most of the literature that I've seen on insulators is it doesn't always work in all species, so it's something that you have to test out, but that is another, you can use specific intervening sequences to damp down that enhancement from 35S. So.
Again, please remember to give your name and affiliation.
My name is Kelly Chapman, and I'm actually a dinosaur in this. I worked in it many years ago when gene silencing wasn't even understood yet, and I also missed part of your beginning presentations. I'll apologize if this is a bad question, but it looks like to me that there haven't been a lot of changes in the promoters that are being used in many years. It does sound like there's a much better understanding of those. Are there many new opportunities coming for promoters and terminators that weren't used in the past? And my second question would be, would any of you like to speak at all to regulatory? It was mentioned one time, but backbone, vector backbone and selectable markers that have antibiotic resistance and so forth associated with them are a huge issue if you are in a global regulatory space. So I don't know if you'd like to address that at all.
I'd like to suggest something on the first part of your comment, is that I really think we need a resource for what promoters are out there, what's available, what do they do, and terminators as well. And I'm going to volunteer PlantGene to be that resource and have it on our website, because I don't know how many people have come to me and said, Joyce, do you know about a fruit-specific promoter, or do you know about a promoter? And we have also timing of expression. So I don't know if I'm exactly answering your question, because as far as what new promoters out there, yes, there are. But unless you go into the literature and take the time to do that, I think what we should have is a resource that says, here's a promoter, this is what it does, some other information that you can go to, kind of like a one-stop shop for that information. So I think we should do it. So I think we'll have Amy reach out to the community, and if you and get that information, and we'll put that resource up, and we can continue to build on it as new promoters come available and terminators. And you want to address any of the other comments?
Regulatory, it's a whole topic by itself. So I think I would advise us to stay on the topic, but we can talk about that offline. But it's a very important topic, but many times, most of the work, a lot of things don't see the light of, going to the consumers, right? And so what we are focusing today is R&D, but it's very important, very relevant, and then things are changing, or policies are changing quite a bit in that landscape. Thank you for question, very important.
Hi, Gina Newman from Solus Agrosciences here in St. Louis. As Joyce said, it would be great if we had kind of a validated parts list. I'm wondering if anybody has good resources for where you go for source of truth for various different versions of 35S. It would be great if we had some shared resources, but if you have any tips and strategies for kind of making and creating your own annotations and what those true sequences are.
Good question, and testing is the only way to the truth. Unfortunately, because what is true to me may not be same for you, because it is true for my own setting and parameters. And so unfortunately, that is how it is. But talking to the people who have worked in this field, that may be the best approach to follow and pursue.
I have a question. So I'm Prasanna from Clemson University, and so when we work with all these vectors and all those things, we would have got some personal addition to it, like do this, like when I used to do the protocol says this, but I tweaked it a little bit. So like, can each one of you or somebody can say some tweaks which is working for you, but we don't put it on a paper, or something like that, which might really help. So can you guys have any tweaks which you do, which has become a part of your vector design and all those things, but we normally don't put that on a paper or something? It'll be like, I add a pinch of salt or extra when I cook, something like that, kind of thing. Or
Extra love. Don't go to coffee while doing the experiment. Finish the experiment, then go, or if you're preparing. But anyways, I was just making it up. Bill, Nigel, everybody, if you want to say something. But thank you for that question.
Yeah. So I think that's we recognize that there's a gap between what people publish in the literature. The protocols can be very terse. It can be very hard to dig back through the literature to find out, they'll refer to a paper that refers to a paper that refers to a paper. It's really hard to get all of that information, and people always leave things out. You try to put in everything that's important, but you have a size limit for what you can put in a manuscript. So part of the purpose of PlantGene is to try to foster more communication about all those tips and tricks. So the interactive workshop on tomato transformation that was held, and people are finding this very useful, and it's the kind of thing where we need more feedback on, what topics can we address through, these types of virtual workshops to help people learn all these tips.
Nigel, you want to say something?
I would say so from the cautionary side. So because of this reason, people tweak things, and sometimes mutations, things happen, as these things have been cultured or they've been stored in a minus 80 for however long, and somebody took them out, they put them on the whatever. So when someone sends you something, oh, we use this, or you find something, your supervisor said, yeah, there was a guy in the lab and he did this stuff, and back in 2010, go and find the plasmid. You've got to be really careful of that, what you're getting when people send you stuff. So either now it's really easy to send it away, sequence it, do the quality control, or go back to a source, a commercial source, and get something that is certified quality control and you know is good. So I think probably lots of people in this room have suffered from that, taking something that somebody gave you and it doesn't work, or it doesn't work the way that you want it to or they said it would.
One comment I would like to make that helped me enhance the efficiency of the transformation. When you're preparing an explant, so it needed to be healthy, it should be healthy, and then when you cut it and wound, don't let it sit for very long because it generates the defense response, wounding response. So if it stays too long, then those cells will have enhanced defense response, so efficiency can become lower. But how long or that wait time is, so minimize that as much as you can and do it in quick batches, so it will help. So that was the tip. You cannot say 5 minutes, we do try to write it, but there are some tricks to it. But watch what you're doing, do the controls and things like that.
Okay. Yeah, so I just wanted to build a bit on, there was a mention of our interactive series. So if you're a Plant Gene member or not, but you've been following along with us, we had these workshop formats. And recently, just to follow up, we have these interactive sessions where you get to have one-on-one through Zoom. It's like coming to the person's lab and having that access to them. And our first one was on tomato transformation. So my group met with people, we went through a protocol, and we had a follow-up session. And this was successful, so we thought, all right, let's continue to go on. So if there are other, like you said about these nuances, and that's kind of where this comes out. And so when we send around a survey and we ask for suggestions, please provide those suggestions because we want to do this. And our next interactive session, which I'll talk a bit about later, will be on agrobacterium preparation. And then we've heard from the community, you want to know about rice transformation and then soybean transformation. So we're going to continue on that, maybe not have so many workshops, but then have these interactive series where those kind of items can be addressed. So great. Good.
Thank you so much.
Sure. Okay, so I'm going to lump the next two together. We're talking about enhancers in the promoter. Often you'll use an enhancer, but to be aware that this is kind of like this be aware. This is sort of the theme throughout this presentation, is that it can have affect expression to other genes in the vector. So don't think of all your vector components as little islands or they don't interact. There's this continuation of effect that can happen through your vector. And avoid using the same sequences or elements multiple times in the tDNA, and again, can be a problem, be aware. Anybody want to build on that information from your experiences or some things to bring to people's attention about these enhancers? Are you using a lot of enhancers? Sometimes I see people do, some people don't. Question over there.
So I am Francesco Capai from Aferna Bio. I have a question more than a comment. So also following up on what some other people asked, what is everybody's golden standard in terms of promoter, enhancers, and terminators, let's say in Arabidopsis? Is there maybe a consensus for, I should say, stable constitutive expression of your gene of interest?
So it's going to depend on, what you need for your product. But for Arabidopsis, there are some well-characterized Arabidopsis ubiquitin promoters that, tend to have different levels, different strengths of expression. So you can kind of, pick the ubiquitin promoter that you're interested in, depending on if you want really high or, kind of a little bit lower expression. But also keep in mind, if you're using these promoters in other crops, it really becomes empirical. You really just need to test it out.
Any other comments? Anybody sitting at the table or in the audience? Did you? Okay. Sorry, we're recording, so.
Hi, I'm Susanna. I work at CTC with sugarcane. And I have a question about going beyond promoters and terminators, because what if you're having very good mRNA expression and you're not seeing silencing, but you don't have good protein expression? What are some tricks you could use to fix that?
Anybody here? Anybody out there have any tips for Susanna?
So, I think, one thing that kind of pops to mind for me is just thinking about protein stability. And there are ways, if you look at literature for the end rule, there are modifications that you can make to the amino acids near the front of the protein, because the amino acids in a protein can get chewed off. And so your methionine tends to be a stable amino acid. It has it imparts a long half-life to the protein, but there are other amino acids that are very short half-life, and it can reduce your half-life of the protein from a couple days to a half hour. And so, if you want to use that type of mechanism to control the half-life of the protein, there are some really slick ways. You can put a ubiquitination sequence on the front of your protein so that when the ubiquitination sequence is cleaved off, it leaves behind the amino acid that you're interested in controlling the half-life of the protein. So some pretty neat tricks, but also, something to keep in mind when you're looking at the amino acids at the beginning of the sequence.
So based on the literature, I want to add a little bit on the terminator side, which is a 3' UTR. So if you use if you're using same promoter, but you are using different 3' UTRs, that also affect mRNA stability and also translation efficiency. So that's the different considerations, not only the promoter, but also 3' UTR. You can modify the gene expression level.
And my input, and feel free to correct me, but I believe codon optimization could also be a potential, yeah, it looks like you've considered it, never mind.
But a good reminder, Katie. That's good.
Hello, I'm Caitlin Vondracek from the University of Florida, and I actually had a follow-up question for your comment about codon optimization. So does anyone here have experience with codon optimizing a vector, and then did it work? Did it have a significant impact on your efficiency? And do you have any kind of strategies or tips for people who are looking into codon optimizing for their own species?
I see some heads shaking, so I think there's some comments that could be made on this.
So yeah, I can start off on that. We codon optimize for different crops that we work with in Corteva, and in general, you can kind of use kind of a general rule of thumb that monocot codon optimization is going to have a higher GC content overall than dicot. It's been kind of interesting to me that that dicots are more accepting of monocot codon usage, and you go the other way, and the maize doesn't like an Arabidopsis sequence, for example. So, so someone also mentioned, and this is something else to consider, let's say you want to express, 3 different proteins in your tDNA, and they're all hooked up to the same activation domain, okay? The sequence that encodes that activation domain, you've repeated it 3 times. That's going to cause problems in terms of stability in Agrobacterium. A lot of times repeats will start to get looped out. Your tDNA becomes much less stable. It can also cause problems in the plant with silencing if you have repeat elements. But so how do you deal with that? Well, you can, even without going beyond your crop's tolerance for its favorite, kind of range of codons, you can change the wobble base on the 3rd codon in those 3 activators. You change the sequence enough that it's no longer repetitive. Agrobacterium doesn't see it as being repetitive, but it's still coding the same amino acid sequence. So there are a lot of tricks you can start to play with that. Codon alterations and codon optimization can be really important in your construct.
Great information, Bill. Thanks. I have also experienced that dicots are a little bit more accepting compared to monocots with a particular sequence.
So my name is Bobby. I'm a graduate student from Purdue University, and I guess kind of like a comment, maybe not the makeup of like your plasmid, but I know for Arabidopsis, tDNA is used a lot of times for generating mutants of the genes that you're interested in. And what I've learned in my PhD career is that sometimes when you have tDNA insertional fragments of similar sequence homology, it can lead to, when you have multiple of those tDNA fragments in the same background, it can lead to heterochromatization and splice out intronic tDNA insertional fragments that can lead to transient expression of genes that you thought were knocked out. So I was wondering, does anybody have anything to say about, like, any comments about that? Any experiences with that?
That's a good comment. Anybody?
I do want to say I concur. That's all. It's like this, that happens. And only way to think about is like generate more events, find the quality events, focus your energy towards identifying those and work on those rather than, 2 types of work I can see, like somebody who is working on improvement of the transformation process and methodology, and then the other group of people is I work on the trait. And so if you are working on the trait, you belong to that group, generate more events and work on quality events. If you belong in the other group, there's so much to explore, and it's fascinating world. So both sides are exciting. And there is NSF people here, and I say, give more funding. That's all I can say.
Where'd she go? Okay, great. All right, good. Let's move on to terminators. We've talked a bit about terminators, and we have this little icon here about knowledge gap, and you can tell by the size of the font and the space on the slide that that backs it up as far as terminators go. But as we mentioned before, terminators can make a difference, so don't just ignore it. It's in a vector. Maybe pop it out, put another one in. It's not a stop sign, right? So it's not terminating. You can have, it's just a slowdown. You can actually have read through your terminators. And because of that, it can affect the final expression of its own gene that it's terminating, but also the next gene in the cassette. And we've talked a little bit about that. Does anybody want to comment on it anymore? Just be aware. But again, a knowledge gap, and we do this by learning. And I don't even know if there's any really publications that address this, but it's interesting to know. But again, I think we need to know what terminators are available and have some information on our site. All right.
So
Okay.
we were talking about this particular topic yesterday, and it's clearly something where more information is needed in the community. So we've talked about read-through and that type of thing, but we've done quite a bit of comparisons within Corteva where we have a trait gene that we're interested in expressing. We use the same promoter, but we use 6 different terminators. And you can get differences in expression levels that are just astonishing, from weak all the way up to really high, just by changing the terminator, with no other expression cassettes nearby. So the terminator can have a big impact on the promoter in that cassette. And we need to get more information out to the research community on this. I think plant gene might be a great way to help with that. And we've got a lot of this data that I hadn't even thought about how useful this could be to everybody. But maybe in Corteva we should think about how to. I'm looking at you, Brian. how we could get this information out the door.
But one thing is knowing that terminator matters and impact. It is a good starting point when you are experiencing a issue. So many times what Corteva did, you have to make an observation in your system. So even if you don't have that accessible information, knowing that matters is very important knowledge to have.
Yeah, checking, if you're interested in expressing higher level of proteins, checking the mRNA or transcript level may not be sufficient. So you need to have some kind of control to measure that.
Oh, I was just going to ask Corteva, yes, please share the information. It would be really useful. And yes, you still have to go and check empirically in your own system. But I can't remember how many, is it 350 years shared experience we have?
170. Don't double it. Not yet.
And we had exactly this, as Bill said, conversation yesterday afternoon, or yesterday morning, is that we all know it's important, but we don't really know which combinations are best or what to do or what not to do. So that is a gap in the community for this piece, and it's important.
Okay. And we mentioned knowledge gaps. It gives me another plug for Plant Gene. We have on the site, what we heard from the community was, how do I find collaborators? How do I find collaborators within academia, public and private sector? So we started a collaboration hub, and this terminator knowledge gap reminded me of that. So if you go on and you log in through our member portal, and there's a collaboration hub, and you can post an idea, you're looking for collaborators, you want the funding, and I think that's a great way to help fill in some of this knowledge gap. And it's both on research but also on publications. So let's use that and try to address some of these questions.
So Joyce, funding, Plant Gene gives the funding?
No, we don't give funding. No,
Okay.
no. Did I say funding? No.
Yeah.
You got to go to other people for that. So,
Okay.
Margaret has a question for someone.
Margaret, can you introduce yourself?
Okay, Margaret Young, Elizabeth City State University. I know we do a lot of mixing and matching of promoters and terminators, bacterial, plant, virus. Anybody have any idea as to what's best? Both plants, whether mix and match is best?
Anybody look at that comparison?
So question, what can you repeat your question?
Like we have 35S promoter from virus, and then we have the NOS terminator from agro. And I'm just wondering, are other promoters from plants, and then we use like NOS or some other terminator, Are there any ideas as to whether it's best to do the promoter and the terminator from the same organism, or it's still best to mix and match them? Any ideas?
Good question.
So there are sequences that we know can cause some problems. I think 35S is probably the best characterized in this regard, is that there have been examples where it can cause silencing in subsequent generations. The NOS promoter and the NOS terminator, I haven't heard about that type of information from those sequences. So, I mean, there are a lot of knowledge gaps here, but I think in general, we've got genomic sequence for so many crops now, and I think there's, kind of a trend that, it's going to be more palatable and more acceptable if we can use sequences from the same crop, just mixed and matched in different ways. So I think, that's something about, something to consider also. If an intragenic mixture of promoter, structural gene, and terminator would be more acceptable to the public than putting viral sequences in the plant, for example.
Bill, back to your discussion about the promoter-terminator interaction. Are you aware of, outside of Corteva, anybody that's working on what the cellular mechanism is that's causing that situation to occur?
Well, there's been a lot of basic work that's been done on the mechanism of eukaryotic terminator sequences kind of looping back onto the promoter to help in the recruitment of transcriptional machinery. So there might be something to that is involved in this, but I don't know that people have really systematically looked at different terminators to see how they're impacting that, or if that's even the way this is occurring. Anyone else have any comments on that?
Hi, I'm Greg Gorologia. I'm from Oregon State University, postdoc. I just want to say that there's, on the mRNA stability issue between promoter-terminator pairs, I forget some of the exact mechanisms, but there's a fantastic paper, I think in Nature Plants 2 years ago, from Christine Quaich's group at University of Washington, and they looked at, I think, 3,000 terminators from maize and Arabidopsis, all with the same promoter. Now, it's not stable transgenics, it's just transient, but that can be a good resource as a library of terminators as a starting point for some people. So I've personally grabbed a few from there, and they seem to work fine, at least in like tobacco leaf infiltration. So that's it.
I was hoping you'd have a comment. Thanks for sharing that.
Great. Anna?
Hey, thanks for bringing that up about the transient versus stable. I'm wondering how accurate measurements of these promoter, terminator, and other elements can be when you do them in a transient system. How well do they translate to a stable system?
I'll bet there are a lot of people who have experience with that, right?
I would like Kiran Mysore to comment on that. He did postdoc. He's a professor at Oklahoma State University, and then he did postdoc with Stan Galvin.
Can you repeat the question, please? I was not listening.
Are you sleeping, Karen?
So transient versus stable, if you have a high transient transformation efficiency, can it be translated to stable? And then if it is not, what could be the reasons? Is that fair? So can you use the transient transformation efficiency to tell you if stable will work?
Yeah, that's a good question. No, it doesn't always correlate. So for many plant species, you can transiently transform, but it's very difficult to get a stable transformation. So transient doesn't always correlate with stable.
And what could be the reason, Kiran? Little bit. You have done a lot of work in this
it's known
field.
that the tDNA can become double-stranded in the nucleus, and it can start expressing even without integrating into the chromosome. So we still don't know how it becomes double-stranded and it can start expressing, but it does. So and it can stay for a while, but once the cell starts to divide, it will be lost. So yeah, the tDNA can express without integrating into the chromosome. It can express. So that's transient expression. So transient expression doesn't always correlate with the stable.
I think you can use transient to tell you what will not work in the plants. So you can help screen things out, but if it is working transiently, it does not mean it's going to be good for you in a stable scenario.
And it may not be bad because if it expresses transiently, you may do the edit and it doesn't get integrated. So there is, again, give and take in anything we do.
I'll actually add to that a little bit too, because when I was working on my thesis project, the first thing I had to do was find out, was my DNA even getting into my explant material? And so that actually helped me by looking at transient. I was kind of identifying what was the best explant material for me to transform. And once I identified my higher transient expression, I was then able to go on and get stable integration in teosinte.
Anybody else from the audience want to comment? Like we as a plant gene would like, community member, there's so much wealth of information right there. So please jump in if you want to make a comment. One more comment.
Actually, I think we should move on. But thank you, Veena, for putting everybody in shape. Okay. All right. So we're going to bring it back to agrobacterium again, because you're like, why are you talking about agrobacterium? You have to take it into consideration when you're choosing it, when you're doing your vectors. So we we have listed here these different strains, and they're all versions of the same strain. We can talk a bit about that more. Again, this be aware comes up again, that strains have been shared. And I always like to use the analogy of a sourdough starter. I do sourdough. So you might get a sourdough starter from someone, and then you take it, and then you work with it, and then you give it to someone else, and there can be changes. There can be other microbes that have been introduced, and it's just something to think about, especially when you're like, oh, well, my transformation was working perfectly fine when I started, and now it's not. And I can give you a personal example of that. Is we received agrobacterium that had a vector in it, a construct in it, and we were told to use for a potato transformation, which was pretty routine in our lab. This is with Desiree. We couldn't get it to work. And I won't go into a lot of the detail, but I said, well, maybe there's a contaminant. Maybe it really isn't agrobacterium. So we did something called a Benedict solution, where you flood your plates, and then you look for a color change. And sure enough, it was not we could see a little bit of agrobacterium, but it was primarily a contaminant. So it's good to be aware, especially, like I said, when you start. I see a lot of head shaking out there, right? It's kind of interesting that you think of more of the complicated things, but that is something to be aware of there. Any comments on? Oh, looks like Consub's going for the microphone.
I want to add that in the community of agrobacterium, the C58 has been widely used, and it was a reference genome, the first sequenced reference genome, and it was only a few years ago when there are different types of strains. One, it has the wild type, original wild type C58 strain has 4 replicons, 1 circular chromosome and 1 linear chromosome, and AT plasmid and TI plasmid. And I believed I was using the wild type C58 until like 2 years ago. we discovered not only our group, but Clay Fuqua group from Indiana University, they discovered that some agro C50 strain has fused the linear chromosome. There's no circular chromosome. It's a combined, so there's a homologous recombination. So there's a large linear chromosome, and then AT plasmid also has a large truncation. So its size was reduced by about 40%, and there's a TI plasmid. So TI plasmid looks intact, but other part of the chromosome is totally rearranged. But we label them C58. I have been using for more than 10 years without knowing that it's a mutant version. Another instance is AGL1. Our lab has our own collection of AGL1. I believe it's the same as other AGL1 strain. I made a thymidine oxotropic strain, and with Arabidopsis infection, it looks okay. But when we tried for maize transformation, it was not working at all. So that was shocking. So I contacted other colleagues, including Joyce and Stan, and they sent me their strain, and we tested it. They are different. I tested 3 AGL1 strains. They all behave differently. So be aware. This is another be aware. So if you're really interested in using a certain strain that you are not using right now, make sure you get it from a reliable source. So that will be critical.
All right. Let's go on to the next item. Oops, sorry. Oh, sorry. There's a question over.
So I'm Ermin Lai from Academia Sinica, Taiwan. Actually, I'm working on aquapotassium for 30 years now. So basically, it's like say even the C58, different source of C58, they are different. So we recently also sequenced our own C58. So there are a lot of variation compared to the published one. And for all those strains, which orally the background is from the C58, but actually the background strain, the cure strain actually is different background. For example, GV311 is from suppose C50C1, but the background EH actually is from A136. So it's a curing by different group. So we actually also sequence those DSOM strains. The sequence is available in NCBI. And then we found there are a lot of deletion, and not only also SNP. So I think that's something that you need to be aware. Even they are all from C58 background, the sequence actually are quite different. Yeah. And another comment about the stable transformation and transient transformation, we had experience using our dropset. For example, we have the seedling transient transformation. The strain worked very well, but actually worked very poorly in for deep transformation. So I think you really cannot really guarantee the correlation with transient and stable. And especially, you also need to consider the tissue specificity, because some are in the somatic cell, and then the other method is in the germ cell. Yeah, just share my comments.
No, great comments. Thank you. That's really good information.
Hi, I'm Antawi, a postdoctoral associate from the University of Florida. There are many cloning experts here, and we are talking about cloning technology and also transformation. So I saw, Katie mentioned, I think Gibson assembly, and there are also another powerful cloning technology, K-way technology. So what I am curious is, first we, use the ADD, PP reaction. So we add the ADD side to the PCR fragment, and then we got the product in the entry vector, and then we do the LR reaction. So what I am curious is, why don't we attach ADDL to the PCR fragment, and then do the LR reaction? Then we can skip one more step. So that is what I'm curious, and I would like to discuss with the cloning expert in this room.
Does anyone want to comment on that? I don't have any experience with that. Or maybe if you want to find someone later and pick their brains about that, I think that would be good as well.
Okay, thank you.
Okay, great. But yeah, thank you. Thank you for the comments. Okay, all right. All right, so we heard a bit about, the origin of replication and its compatibility and affecting copy number from consub. So I think we've got that pretty well covered. And then...
I actually had a question about that.
Oh, it's you. Okay.
Yeah, so the copy number in the agro, is that going to affect the copy number in the plant that gets an integration? Is there a relationship? Because we don't always want multi-copies in, actually we don't want multi-copies in the plant.
There was some older publication from 2009 and 2010, a collaboration between Stan Galvin and my supervisor, Kan Wang. They tested some extreme cases. So chromosomal launching of tDNA to the plant, and also from high copy number of plasmid, PVS1, and also low copy PSA backbone. So there was a correlation. So plasmid transmission frequency and also plasmid copy number, they are correlated. So in general, high copy number resulted in the high copy number plasmid usually result in higher transmission frequency, but at the expense of usually in general higher tDNA copy number per cell. So there is a trade-off for sure.
Right. Okay. Next topic has to do with inducing the Vir gene system, and as many of you probably have used acetylsyringone to do that. But we wanted to, again, just be aware is that that's not always necessary, and there can also be negative effects. I've experienced that myself where it really didn't make a difference. But in some plants, it can be plant species dependent. As we were discussing yesterday, I primarily work with species in the Solanaceae. They give off a good number of phenolics that induce that Vir gene system. But for other systems, you may have to go through some sort of protocol change to induce that Vir gene method. And do any of you want to kind of talk about the sort of approaches that can be used?
One thing to add to your comment is some strains have constitutive expression of virulence. So know your strain. That's all I would like to add. Bill, or somebody else would like to add more? Very important consideration. And then if virulence genes are not induced, tDNA will not be processed. So you think you are doing something, but actually agrobacterium is not doing things. You have question? No? Okay.
Yeah. And again, we just recommend that you do a comparison, right? Use it, don't use it. I mean, some people put acetylsyringone in the co-cultivation medium, but that can have a negative effect on your plant regeneration. So just, again, I think just a question, do I really need this? Do I really need this to happen?
And I never thought acetylsyringone can go bad, but it can. And so make sure you are using functional reagents.
A fresh stock.
Fresh stocks.
Yeah, exactly. We make ours up fresh when we need it. Okay? All right. And then we have here this kind of idea of rearrangements that can happen when you put your construct in. I haven't experienced this often, but it has happened where we do a transformation, we don't know what's happening, because we might do the checking in E. coli, but not in agrobacterium. It's fine in E. coli, but then when it goes through the agrobacterium, we then see a rearrangement. Sometimes it's the more complicated type of constructs that we put in. And this rearrangement then can result in you not getting the results that you want. And to be aware that there are these recombinase positive and recombinase negative strains that, that you could use, like the AGL1 we mentioned. That's actually become a favorite in my lab for this. A lot of the reasons because it's recombinase minus, but to be aware of that situation, especially if you're not getting good results. Any other comments on that, about the strains?
One comment here is we are doing more CRISPR-Cas9 So there are recombinases in the vector, so it makes it more common observation compared to conventional vector strategy in the past. And then we are putting a lot of cassettes in a tDNA also. That causes additional like things to think about when there is a stability issue.
Yeah, I'm Hejin Kim in the Bayer Crop Science. So I had the same experience when I was postdoc in the UNL, the University of Nebraska Lincoln. One of our project introduced 13 cassettes. The vector size is that more than 30 kb. So the issue was, I think, it's not the repeated lesion, but it's the size. So bigger size always give the difficulty of the cloning, and also is that the rearrangement of the agros. And I keep failing when I use the GV3101 at the point, but tested a lot of agro strains, and finally found out the Legge minus strain, and it is stable in that. So, and also it's not always all colonies are stable. Some colonies still making some rearrangement. So I really appreciate to recommend to the agro QC for that by the whole plasmid sequencing in agro is that to see that the stability issues. And also the one recommendation, we covered a lot of that the promoter terminator, but I wanted to talk about the intron too, is that influence a lot of the expression of the gene of interest. So it's when you introduce the intron into the CDS region, it's affect a lot of the gene expression that is considered to make that vectors.
Yeah, very good point. Very good point. Okay. All right. If there aren't any, oh, just one there, Kieran.
This is Kiran Mysore again from Oklahoma State University. I was under an impression that all AGL1 strains available are all RECA minus. Am I wrong? Are there RECA plus and RECA minus AGL1 strains?
Yeah, all AGL1 are Reggae minus.
Okay, how about EHA105?
EHA105, we have a Reggae minus strain called EHA105TR.
Oh, okay. But all AGL strains are RECA minus, right? AGL1
No. The AGL1 is generated from AGL0. AGL0 is not, is Reggae positive, but AGL1 is Reggae minus.
Minus, okay.
Yeah.
Got it. Thank you.
All right, anything else? Okay, so next slide, and we heard a comment about using introns to help your gene expression, but also using introns so that you don't have expression in the agrobacterium. And an example of this is, let's say you're using a reporter gene such as GUS or GFP, and you don't have an intron in there, and you look at your material somewhat shortly after doing the transformation, you think, woohoo, successful, I see expression, but it could actually be coming off the agrobacterium. We've experienced this as well. But if you use a reporter gene that has an intron in it, you can, that is a better indication that you're actually seeing the expression coming off the plant cells and not from the agrobacterium. Any comments about that from anyone? No?
So when you do get this kind of spurious and unexpected expression of your plant cassette in the agrobacterium, agro will not recognize all plant promoters, but some promoters work pretty well in agro. And a gene that you expect is going to be perfectly benign in the plant, for some unknown reason, might be toxic to your agro. So it can really throw a wrench in things that's hard to understand if you're not paying attention.
Yeah, we've had that issue when we were trying to express an iron and zinc transporter. We had real problems with rearrangements in LBA 4404, but when we switched to AGL1, it was okay. So, I mean, just another story. Check your agro. We've had, before, we do it every time, just when we're doing the co-culture, for the first time with a new gene construct, we take a sample, and then we sequence that. We've had cases where it's stable if you're growing your agro on the plate, but if you do an overnight liquid, all goes to pieces. So you think it's stable, it's not always. So we check it right when we're actually using that co-culture media for the inoculation medium.
Okay, these next ones on the list here, kind of agrobacterium trivia. This was Wayne's. So I think we all had to write down the answers because only Wayne knew. So who knows what LBA stands for and LBA 44 are for?
You have to give the right answer.
I'm sorry, there are no prizes. but you have to get the right answer. Anyone know? Okay. Vina, do you have the Wayne's email? Okay, LB is explained in the original paper, the Latin name for Leiden. So if you want to look up the original paper, and then he has a whole thing here on, oh boy, the name Lugdunum Baltivarum during the Renaissance. Anyway, wish Wayne was here, he could give us a little more background. But there is a reason for it, but it's a little more historical and something about being part of the 15th, 16th centuries, and anyway, look it up. All right, so the next one is GV31. What's the GV stand for? Nobody knows? Okay, well, that's okay.
Grape wines.
That's a good guess, but you're not a winner. So GV was also in an early paper, Genetisch, is a German virology, and they did the agro work in a virology lab. So it's a genetics virology lab, and it was called GV. And the last, we're doing pretty good here. So the last one is EHA. What does that stand for? Kieran, good. Go ahead. Wait till you.
Elizabeth Hood, I think she was a graduate student with Meridel Chilton, and she's the one who developed these strains, so it's named after her.
Great, good, okay. Maybe we'll give you a plant gene member pin. Woohoo, okay. All right, well, that was just a little bit of fun. We're getting near the end here, guys. The next thing is that we had talked about maybe including agrobacterium preparation here, but we're already close to 11:30. But we decided this would be our next interactive series, an interactive session in our series, and we will announce that in the Plant Gene email system. So that will be, again, meeting with the group, this group here and others, and having this conversation, kind of like you're coming together with somebody in the lab and talking about, kind of best practices for agrobacterium preparation. So keep an eye out for that announcement. And then we felt it was important to talk, a little bit of quality control information has come up, but to keep in mind best practices when it comes to your cloning. Remember, plant transformation takes a long time, right? From start to finish, some species longer than others. It's so worth it putting in the time in the beginning to save yourself time and resources at the end. So if you don't take away anything from this workshop, just please remember that quality control is so important. And that could be, digesting and or sequencing your construct before you do a transformation. Check when E. coli and check when in agrobacterium. It's really, we talked about rearrangements. And again, before you go through that long journey of doing your plant transformation, put in that extra work. And then, we have some information here about your cloning method and that your approach and application for your intended design. So just, again, be really, really good practices and put in that work ahead of time. Anybody want to share any experiences with this, quality control, any suggestions for doing this? Yep, back there.
So I've used the CRISPR-Cas9 system to try to get some mutants in Arabidopsis, and one of the mistakes I made when I first started was I did not get rid of the Cas9 from the next few generations. So I actually got more edits at my sgRNA site than I wanted, and it turns out I had a 3 3 base pair deletion. So I deleted 1 amino acid and nothing happened.
Well, thank you for sharing that. That's really good, and some people can learn from that. It's helpful when you share those pitfalls that you've learned. So, over there, Greg.
Hello, I'm Luisa Bermudez for Gali Company in Boston. Normally, what we are doing after the checking agrobacterium is to make an agroinfiltration in Nicotiana pentamiana just to check that it's working. I don't know if you are normally doing that also as quality control, or if there is, I don't know, any other option for that.
Yeah, so infiltration into your plant material and looking at it that way. That's good expression. Okay, and then Greg over there.
Hi again, Greg. Greg here. So one thing I was going to mention also as a pitfall is I have kind of a psychosis with over-engineering my stuff. And now that you can order synthetic fragments, it's almost a problem because the stuff that's most over-engineered, I find is very unstable in E. coli, and I can't recover the original clones. And so just, of course, do all of your design, but the synthetic fragments that you order are often not going to be stable in E. coli unless you have something where they're going to deliver you the E. coli to you. So that could be a reason to go that route versus like a gene block. So.
Sabine and I were just discussing, and again, another plug for Plant Gene. We have a discussion forum. And that discussion forum is based on email. And you send an email, and it goes out to the community. You have to sign up for this discussion forum, but if there is anything else that you've thought of after this session, and you want to gain feedback from the community, it's a great way. Send an email. We've got it working. You can get the information on the website on how to sign up. Go to that member login portal. And you just send an email and somebody will respond. And we've had some really great comments. I've used a discussion forum before to find out about LED lights. What are people using? And got great feedback. And so we really encourage you to sign up for that discussion forum. And if you have other questions or comments that you want to share, that's a great place to do it. Okay, great, good. And we're at the end. And I just wanted to tell you that next year, 2025, again, we're building on this cloning theme, and we'll be doing modular cloning systems next year, and we really hope that you'll attend. We'll get the word out about that. I want to thank all our speakers, the panelists here, all of you for your participation. This was great. I hope you found it useful. A reminder that the recordings will be on the SIVB website, and then there's a contract that we have to wait till it's released. And then it will be on the Plant Gene website for as long as we can, and the Plant Gene website is hosted on SIVB. And remember, as I mentioned in the beginning, if you're a Plant Gene member, we have these cool pins. Be part of the cool crowd. Come up and get your Plant Gene member pin. And any of the panelists here want to add any comments? I knew you would have a comment, Amy.
I want to add, because Prasanna reminded me one more thing, that please give us your feedback, how you like, what you like to see more in future, and something it did not work. So we look forward to your feedback to get better and then have a good resource for community.
Yeah, I agree. As we say, the Plant Gene is a community-driven initiative, and that means we're getting input from you. It's like, what do you want? What would be helpful? That's how this came about. Some of you might have found this kind of basic, but we felt that for those who really needed the basics and needed to know how, and we've heard like why. We're told, oh, don't do this or do this, but why is that? And unfortunately, the generation coming up, they're just not getting that in classes. There used to be classes on this and courses, and it's just not there, and we're hoping to fill in that gap of information. So use us and let us know what you want to see. Amy will be sending out a survey for the feedback on this and for suggestions for other workshops, all right? But thank you, really appreciate this. I thought it was fun. And we'll see you around, and come next year to our workshop. Yeah. Thanks.